So I changed my idea on how to handle this data over time. And we will see that there are some open questions that still are open. So first of all, I want to give you some definition. First of all, a metagenome, when I talk about metagenome, I refer to the genetic information that are present in an environment, while microbiota is just the collection of the microbes present in a defined environment. With microbiome, instead, I refer to the totality of microbes and genetic informations, while metagenomics is just the study of genetic material recovered from environmental sample. So one big question is why we use metagenomics. The question is, the answer is that the microbes that we can grow in the lab are less than 1% of all the bacteria that we currently know. So with the metagenomics, we can have almost the totality of the picture. I'm saying almost because it's not completely true. Another big question is how can we study the microbiome composition? So mostly the technology most used right now is 16S or 18S in case of fungi. 16S is defined as amplicon sequencing. So we are just sequencing a small segment of the bacterial genome in this case. The pro of this technology is that it's very cheap right now. The protocols that we have right now for analyzing this type of data are well established, more or less. While the limitation is that we have just an information of the microbiota composition, and we don't have an information on the function, but also we can look only to bacteria or fungi. For viruses, there is right now some kind of technology on amplicon sequencing, but for the moment, with amplicon sequencing, you can just see some specific populations. But why 16S? 16S is the gene of 16S. It's 1,500 pair bases. And it has some regions that are well conserved. You can see here that the mountains are the region that are well conserved in the genome, while the spikes are the region inside the 16S that are most variable. So with amplicon sequencing, we can choose to sequence some specific region. For example, for bacteria, for fecal microbial profiling, usually we use V3, V4 is the most common used because it's short and allows to have a pretty clear distinction between species. The other regions, for example, V1, V2, for example, are used for environmental samples, or for example, V1 can be used to distinguish specific bacterial strains. Another approach is to sequence everything. So shotgun sequencing of all the genetic material that is present in this environment. This technology allows you to have almost the totality of the picture of the bugs that are inside your samples. Gives you also a let's say a functional profiling. So what this bacteria can do. I'm saying potential because it's just the genetic information. We are not talking about proteins, enzymes. That's another story. The problem with this technology, I mean, first of all, is that this technology is quite expensive, so it's not affordable by everyone. And also the pipeline and the workflow that are present right now are not so well established, I would say. Beside all the specific pro and cons of this technology, all the data that we collect from next generation sequencing experiment have some problems that we will see in the next slide. But these problems are compositionality and sparsity. So I will explain first of all compositionality. The compositionality means that the relative abundance of the bacteria that we find is strictly related to the abundance of another bacteria. So I usually make this example. So if we consider a natural environment, so we have in this environment we have tigers and ladybugs. So if in this environment the population of ladybugs increase significantly, the tiger population will be not affected by this increase in the ladybug population. This is called true independence. While in our next generation sequencing experiment, we have the limitation of the technology. Imagine that with this technology, we have a number of slots of species that we can observe. The number of slots are just sequencing depth of the technology. So we need to fill this. If we observe our population, we observe that there are ladybugs and tigers, okay? But if there is an increase in the ladybug population, the probability to observe the tiger population will decrease. This is compositionality. Another problem is sparsity. This means that we have a lot of zeros in our dataset. So we have a lot of bacteria that are present maybe in one or two samples. And also the meaning of the zero in a microbiota experiment is different compared to our other studies, because here zero can mean two things, basically. Zero can mean that this bacteria is not present, or that this bacteria abundance is under the detection level. Okay. Another big problem that we have right now with this technology is that we have a limited number of sequences that we can produce. So, but also the number of sequences that are produced per sample is different. Even though we try to load the exact same quantity of DNA in the sequencing machine, at the end we will have samples with different number of sequences. I will refer to this to sampling effort, sequencing depth, but at the end of the day, we have always these differences in sequence abundance that is not biologically meaningful. Okay. Again, now I think I will talk about the limitation that we have with this technology and some steps that we need to introduce almost in every experiment. Because the biggest problem that we have right now is that every step that we take during our workflow affect our results. Starting from the extraction kit that we use, the PCR, so the primer that we use, the sequencing technology and the bioinformatics, each step add some error inside our analysis. So what we need to do here is to estimate the error that we have in our workflow. We can do it in different ways. The simple way is to use blank samples. So we can use like sterile water, included as treat it as a sample and pass it through each step of the workflow. This will allow us to have an estimation of the contaminant that we have in our workflow. So contaminant can be present, especially in the extraction kits. Another things that we can do is to introduce mock communities and spiking standards. So they do similar things, but at the end of the day, they allow us to estimate the error that we have. So with mock communities, we have a community of bacteria of known composition. Usually, mock community are created based on the matrix that we want to analyze, because sometimes, for example, we can create mock communities for feces or different, for example, for skin bacteria. And also, this composition contains gram-positive and gram-negative bacteria, usually, and we treat it as an external sample and pass it through each step of our workflow. At the end, we will have more or less an extraction efficiency of our protocol. So the only problem here is that we are not considering the composition of our metrics. We are not considering, for example, for feces, for pigs, I'm imagining the fibers contained can interfere with the extraction. So one thing that we can do is to introduce spiking standards. So spiking standards are usually a collection of two bacteria, one gram-positive and one gram-negative, that we include in our samples directly. These bacteria are, alien to the fecal environment, so it's impossible to find this bacteria in the feces. So at the end, these allow us to have a more precise estimation of the error present in our workflow. Okay. I said before that contaminants in the identification is important. Maybe for fecal samples or samples where there is a very high load of bacteria, contaminants are less important. But when you work with samples that have really a low quantity of bacteria or supposed to be sterile, so this I usually are referred to low biomass samples. So for this kind of samples, usually it's very important to estimate which bacteria is a contaminant and which is not a contaminant. So some methods have been developed in the literature. So there is one that is focused on a principle that usually contaminant DNA follow a specific trend. This trend is that when you increase the DNA, usually the contaminant abundance decrease. Okay. So now we move to the most bioinformatic part. And one thing that we should talk about is one of the biggest challenge, the controversy that there is going on right now, that is the introduction of amplicon sequence variant that should replace OTUs. So I should say that this has been was like 2 years ago. There was really researchers that were publishing a lot on this topic, whether or not we should use ASV instead of OTUs. But first of all, we have to understand what is an OTU. So I think this picture is quite representative of what is an OTU. So when we talk about OTU, this refers to operational taxonomic unit. So they are not bacterial species, but these are cluster of sequence with that have a similarity of 97%. When you want to obtain OTU, you start from your community, you cluster sequence based on the similarity, and then for each cluster you select a sequence that is representative of this cluster. For the most part, people use close reference OTU. So this means that you have a data set with the taxonomic information that you can use to assign the taxonomy to the OTU. And the sequence that do not match to this reference will be discarded. Another approach is called open reference. So you align your sequence to the dataset, and the one that do not match are used for de novo clustering. De novo clustering means that the sequence are clustered together, but there is not a reference dataset. So this allows you to include this OTU inside your analysis, but these OTU are specific to your experiment and are not comparable with other studies. So if you want to do taxonomic profiling, closed reference OTU is perfect. But if you want to do more specific things, for example, if you have really a huge data set with a lot of data and you want to train a machine learning algorithm to classify, I don't know, humans based on diseases, for example, Crohn's diseases, usually working with de novo OTUs allows you to have a more precise classification. Okay. On the other hand, new evidence come out, and we move it to amplicon sequence variant, or we should move to amplicon sequence variant. This approach is completely different from the OTUs. So you have your community. What you do here, this pipeline that creates ISV, estimate the amount of error that is present in your sequencing and correct the sequence to align with biological sequence. So at the end of the day, you have a more clear picture of what you have, and the results obtained in your study are completely comparable with other studies. Okay, but now there are some problems with amplicon sequence variant that have been pointed out by Dr. Schloss. For example, one problem with amplicon sequence variant is that bacteria usually don't have one copy of the 6NS gene. They have different copy. And these copies also may have some nucleotides that are different. So with amplicon sequence variant, there is the problem that the genome of a single bacteria can be splitted in different amplicon sequence variant. And this is usually something that we don't want to happen. So at the end of the day, one approach that take the best of the both worlds is to create amplicon sequence variant, but apply some clustering, for example, at genus level. This will address most of the problems. Okay, now we have our beautiful OTUs and amplicon sequence variant. What we can do next? Usually, the typical workflow of a microbiome data is to obtain amplicon sequence variant or OTUs and calculate some metrics. You can calculate alpha diversity, beta diversity, and also another step is to see if some specific bacteria is more abundant in a condition compared to another. And a final step can be to perform functional profile based on 16S. There are some pipelines that can do this, but usually it's pretty inaccurate, let's say. Another things you can do is network analysis So try to understand the relationship within the bacteria inside your community. But usually the most commonly used measures are alpha diversity. So we come back to the tigers and ladybug example to explain what is alpha diversity. So alpha diversity is composed by two things. One is richness and one is evenness. Richness is just the number of different bacterial, the different species that you have in your samples. The evenness is how this population is composed. So in the sense that is there some bacteria that is more abundant, more common compared to another? Some measures like chow are only based on the richness. So just on how many different type of bacteria I have in this sample. Other consider also the evenness. So how these bacteria are distributed. For example, here in this example, richness is always 2 because we have just tigers and ladybugs, and there is a perfect balance in the population. While if you consider the spread in the ladybugs that we talked about, the richness is always the same because we have 2 type of, I would say, animals and insect, but the evenness change. Once you have your data on the alpha diversity, most of the papers that we have treat this data as a parametric data, but these are not parametric data. And they usually do a simple linear model, and maybe they include the sampling effort or number of sequence that I have in the samples in the model to address for the sampling effort problem. Because among all the metrics that we have, alpha diversity is the one that is highly affected by the sampling effort. Another approach is rarefaction that we will look in depth in the following slide. Rarefaction means that we are subsampling our sample to reduce the number of sequences that you have and to allow us to have the same number of sequences for each sample. To do this, we do random subsampling like 100,000 of times. Another thing that I will say that you should not do is to remove rare bacteria from your samples before calculating alpha diversity, because these measures are very sensitive to low abundant species. While beta diversity is another word. Beta diversity means that we are quantifying the similarity or dissimilarity between our samples. We can do it in different ways with different metrics. For example, the most used are Brachyartis. Brachyartis is very common because it's very robust to sparsity. Then there is the Hison distance, that for sure the pronouns is not this. This measure usually is a measure of the Euclidean distance of the abundance data, but transformed using some compositional transformation like CLR. This will address the compositionality bias. Another measure is the unifrac distance that take into account the phylogenetic information in our samples. Unifrac distance can be weighted and unweighted, considering or not the abundance of the amplicon sequence variant or OTUs. Once you have created your fantastic distance metrics, you usually plot it with principal component analysis, or more commonly with non-metric multidimensional scaling. This gives you an idea of how your samples are similar or not. But usually it's just a visual inspection of the data. But if you want to test differences, the most common approach that is well established is to perform a Donis test that usually is an ANOVA model with permutations. Another big point is how do you present your data on beta diversity? Because usually what you find is something like this. A plot with clouds of points, and if you're lucky, the points are well separated and the separation is clear. But in other points, in other situation, maybe the points are not so, there's no clear pattern. And when you present your data, you say, oh, okay, I see that this treatment, pigs receiving this treatment have a completely different microbiome compared to the control group. And then you have a cloud of points that in some way they are overlapping. There is no distinct pattern. So what you can do here is to use another kind of visualization of the data. I will not use a bar plot, but anyway, box plot are better in this case. So you can see this is another study, and you have a representation that is different. They tell the same thing. So the bar, if the bar is very high, you have more differences inside this group, and if it's low, you have less differences. But this approach maybe can help us to explain our results. Okay, another important step is to test differences in the abundance of specific bacteria. Over the years, they've been developed, I don't know, I didn't count it, but several methods to do this. So here, what we want to see is the differences in the abundance of a specific bacteria, specific taxa. The method that you use in this case will define your results. And that's the biggest problem, because depending on the type of normalization of your data, depending on the tool that you use, you will have a different result. But for example, I really suggest you to read this paper. They reviewed all the methods that are present at the moment across different datasets. All the methods that are used, for example, rely on parametric tests, for example, t-test, ANOVA, or non-parametric like Kruskal-Wallis. So this first step, I will not encourage you to use it, because they have a big problem, because they have really low power to detect differences in your taxa. So you are missing a big, you can miss a lot of differences if you use classic parametric test. Another approach, for example, is to use another tool that has been developed, is to use linear discriminant analysis and effect size. So you have parametric test, classic parametric test, coupled with an estimation of the effect size of the difference. You usually see it in a lot of paper. Usually all the human studies, I think, have this LEPC analysis, because it's very simple to draw conclusion. It's very simple to present this data, but it has a little problem in the false positive. That is, higher, you can address this by using more strict threshold for the effect size or for the p-values. For example, you can use threshold under 0.01, for example. Other methods, for example, that are completely taken from other fields, for example, RNA-seq. This method have a big problem that is the way of which they handle zeros. Because in an RNA-seq experiment, 0 means a different thing compared to zeros in a microbiome study. Because 0 in an RNA-seq experiment means that this gene is under the detection level. While, as I said before, in a microbiome studies, 0 can mean two different things. One is under the detection level, and one is that this bacteria is not present. Also, that we have other tools, for example, for compositional data. In this review, they tested everything basically, and they suggest to use different methods. So as long as these bacteria come out in different methods, so you can be pretty sure that this bacteria is more present in a group compared to another. And this is the recommendation. While another big topic that I want to discuss right now is how we can normalize this data. Because usually when I start in this field of research, normalization was rarefaction, sorry, was something that to be ashamed of. You should not do it because you are throwing away your data. And usually people, when spend a lot of money on the sequencing, doesn't want to throw away their data. But actually, this is quite complicated. And in the literature, there's been a huge fight, I would say, between two research groups. So now we will see the two sides of this fight. So the first side of this fight was when someone doesn't want you to do rarefaction, usually come out and say, oh, okay, but look at this paper. This is the paper that usually is the most cited when a reviewer doesn't want you to do rarefaction. And usually the comment that people do is, no, you should not do it because you're throwing away some of your data. Because as I told you, with rarefaction, we are reducing the number of sequence in our sample, but by doing some random subsampling. The dimension of the sample that we are creating is dictated by the sample with the lowest depth, with the lowest number of sequence. And people say, okay, but this threshold, so it's arbitrary. But when you select this threshold, you should be aware that there are some cases when you want to throw away some samples that are usually they don't follow the distribution of the sequence. Because usually you can have, for example, the majority of the samples that are around 20K and one sample that is 200 samples. So before doing rarefaction, inspect your data and remove samples that are clearly not following the distribution. So coming back to what people against rarefaction says is that you should use just relative abundance. So relative abundance means that you have the abundance of your bacteria divided by the number of sequences that were present in your samples. So this step is just not enough to normalize your data. What this author suggested instead was to use variance stabilization technique like centered log ratio that are taken from compositional data analysis. Okay. The other side, the dark side of the research field is people who do rarefaction. So this year, another paper came out remaking all the analyses that were performed by the previous paper in question, the famous paper. So here, the author basically took all the dataset and remade the analysis and find some, I will not say error, but misunderstanding, let's say. The big misunderstanding is the difference between rarefaction and rarefying that seem similar but are completely different. Because rarefaction is when you do random subsampling several times, like 100,000 times. This will allow you to have a mean abundance for each bacteria and a standard deviation. While rarefying is completely different. It's doing rarefaction, but only once. Okay, so once the author remade all the analysis, found that rarefaction is still the most robust approach to the different sampling effort. And also point out what I said before, that the choice of the rarefaction threshold should be made critically by looking at the distribution of your data. So I will just show you some results of this paper because I think that these are interesting. For example, here you have all the normalization method, the most commonly used, and their false positive rate. You can see that with rarefaction, when you use alpha diversity, you have a really low false positive rates. While for the other normalization methods like normal relative abundance or compositional method, you have a really high false positive rate, both for the alpha diversity and for the beta diversity. So the problem here is that when you do not perform a rarefaction, you are, let's say, masking the effect of sampling effort into the effect of your experimental trial. So it can happen that for some reason you can obtain more sequence in a group, in an experimental group compared to another. So, and when you see, when you perform the classical normalization method, at the end, you will see that you have differences. But these differences are not driven by your experimental treatment, but the fact that you have more sequence in these samples compared to the others. So when we talk about physis, maybe this is not always the case, because usually we have a number of sequences that usually it's quite similar between samples. So the false positive rate problem is not so high. But if you compare different matrix, like for example, skin microbiota with another mucosa, this can happen that you can obtain a different number of sequence, but not because of your treatment, but just because of the matrix that is different. Okay. Also, another problem is the power of this normalization method. You can see that the power for the richness is very high with rarefaction, while the other diversity measures are really almost not affected in this case. That's because the other measures, like sample Shannon, consider more critically the abundance of low abundant bacteria, while inverse Simpson does also a similar thing, but this takes more into account the presence of more common taxa. So, okay, I think here I left a lot of useful content that you can follow, tutorials, things that can be helpful if you are starting in this field. There is also, I want to suggest you the YouTube channel of Rifomonas project. So the author of the paper that I cited, the Patrick Schloss, he created a series of video on the bioinformatic analysis on microbiome data using R. I think, but they have also other series on other type of data, but it's very helpful. It was helpful to me. And also I left other links to tutorials, to seminars, and other resources that can be helpful if you want to start in this field. That I think if back in the days would help me a lot. So thanks for the attention. I think I covered almost everything. Now, if you have some question, we can discuss. Thanks.
Thanks, Federico. It was a very, very informative talk. So a lot of things you discussed getting the data and analyzing it and then giving us a lot of references. It was a great presentation. Thank you for that. And now I open the floor to questions if there are from our audiences. It seems your talk was very good. No one, you covered everything. No one has
I
any
think they are
questions.
there. No, I think they were striking, but all this information in 1 hour was too much.
but actually, it's needed. I mean, these are the step-by-step things that you need to do for microbiota analysis. But to be honest, I haven't done any of this before, so I don't. I understood what you said, but I don't have any questions myself. But if we don't have any questions from the audience, I'd like to thank you, Federico, for this very, very nice
Thanks.
talk, informative. It's going to be available in the PIGWEB YouTube channel. And for those still here, please stay tuned for the next set of webinar of the PIGWEB Junior Community. Thank you. Have a nice day.
Bye.